Hello friends, welcome to my YouTube channel NEET Biology Made Easy. In this video, I'm going to discuss on carbohydrates. My last two videos were on amino acids and proteins, and you can find the link in the description section of this video. Carbohydrates are one of the most important biomolecules required for the survival of any living organism because they are converted into energy with the help of metabolism. Carbohydrates are widely distributed in plants and animals because plant cell wall is composed of cellulose and exoskeleton of some living arthropods are composed of chitin. They have both structural and metabolic roles in living organisms. They are metabolized into energy. Carbohydrates are the most important and abundant form of biomolecules on earth. And rice, wheat, milk, grains, tubers such as potato, sorghum, turnips are the rich sources of carbohydrates. They are basically classified chemically into two groups, aldoses and ketoses. Here I have shown you the structure of one aldoses and one aldose sugar and one ketone sugar. In these two diagrams, on the left hand side, I have shown you the structure of glucose. It bears CHO, which is nothing but aldehyde group, and that is why uh, this is considered as aldose sugar. On the other hand, you can see the presence of C double bond O group, which is ketone group in fructose. That is why fructose is one of the examples of ketose sugar. Monosaccharides are subdivided into two categories again, based on the number of carbon atom present and also based on the functional group it bears. So, based on the number of carbon it bears, they may be categorized into trioses, tetroses, pentoses, hexoses, etc. And again, we have two possibilities. Either the structure may be a aldehydic group, bond CHO, then they will be considered as aldoses. And if it bears C double bond O, that is ketonic group, then they may be considered as ketoses. Here I have shown you the comparison between glucose and fructose. Glucose bears bond CHO group, aldehydic group, that is why they are considered as aldoses. On the other hand, fructose bears ketonic group, that is why it is considered as ketose sugar. Again, we have two forms of structure, either open chain structure, then Howard ring structure. Here I have shown you how the open chain structure of glucose look like. So let's try to make it by our own. First, you write down CHO because it's a aldose sugar. Then you need to draw all these six carbon atoms. 1, 2, 3, 4, 5, and this is the sixth one, CH2OH. So here this carbon number 2, carbon number 4, and carbon number 5. They are actually anomeric carbon. What are anomers? The valency of carbon is 4. If all these four valencies are occupied with four different functional group, then this particular carbon atom is considered as a chiral carbon. So here uh, carbon number 2, 4, 5 are considered as chiral carbon. So here you just draw OH group on the right hand side on these three chiral carbon atom. Then you have to make just the opposite of it and fill the empty sides with hydrogen. So this is the structure of D-glucose. D means dextro-rotatory, dextro, that is on right hand side. Right hand side, the presence of OH group on the right hand side of anomeric carbons. Then if you just take the mirror image of it, suppose this is the mirror image, then you need to draw the reverse of it only. CHO, this is carbon number 1, 2, 3, 4, this is fifth carbon, and this is the sixth carbon, CH2OH. As we are drawing the mirror image of D-glucose, we are trying to draw L form of glucose. Then on the anomeric carbon, you need to draw the OH group on left hand side, like this. So this is nothing but L-glucose. L means levo. Levo. That means left hand side, on left hand side. So this is how D-glucose and L-glucose the structures can be prepared. Next topic is Fischer versus Haworth projection formula. Here we have the open chain structure of glucose. Here you can see the presence of 6 carbon atom in this structure. So here this oxygen is electronegative in nature, and this carbon is electropositive. And on the fifth carbon position, again this oxygen is electronegative and this hydrogen is electropositive. So if there is an interaction between this first carbon with the fifth carbon position, then this oxygen will bind with this, and there will be interaction between this oxygen with this hydrogen. So you can redraw the structure like this. So here is the oxygen, it interacts with the first carbon. Then here, instead of this double bond, now this will be converted into single bond, and instead of oxygen it will become OH. On left hand side you will have the same hydrogen atom. Then again all these groups will remain as it is. So this structure is called alpha D glucose. Then again, if on the first carbon, if this OH group is on left hand side, and if rest of the things remains as it is, like this, then this structure is considered as beta D glucose. So these two isomers of glucose, where there is only one difference, and that difference is on the position of OH group, whether this is on right hand side or left hand side, and these two structures are called anomers. These two isomers of glucose are called anomers, and this formula is called Fischer projection formula. Next, we'll discuss on Haworth projection formula. The cyclic form of this structure is called Howard projection formula. Let's draw this structure. This structure is easy to draw. So here is the hexagonal ring. This is the oxygen, and all these corners actually represents one carbon atom. These are the bonds. This is the first carbon, second, third carbon, fourth carbon, fifth carbon, and the sixth carbon is here, CH2OH. This is the sixth carbon. So those groups which are present on right hand side of this open chain structure will be on the bottom of the ring, and the groups which are on the left hand side should be on the top of the ring. So the wedge group on 2, 4, and 5th positions are now filled. Then here at the first position, we have to check whether this OH group is on right hand side or whether this is on left hand side. So in this diagram, if OH group is on right hand side in the chain at the first position, then it should be on the bottom, and if this OH group is on left hand side, then you need to draw it at the top. So if this OH group is on right hand side, automatically it becomes alpha D glucose, and if you want to draw beta D glucose structure, then you need to draw this OH group at the top, and you have to put this hydrogen in the bottom. So this structure is simple, and when we will start with disaccharides, this structure will help us to understand how glycosidic bonds are formed. So in this diagram, we have seen that the interaction took place between first carbon and the fifth carbon, and in such kind of interaction, we'll be getting this kind of hexagonal ring where there are five carbon atoms in the ring. You can see there are five carbon atoms in the ring with one oxygen, and these kind of rings are called pyrene. So this structure of glucose may be written as alpha D glucopyranose because it bears one pyrene ring. Then again, if the interaction is taken place between the first carbon and the fourth carbon, then we'll be getting furanose ring. This is the furanose ring. Here you can see there are four carbon in this ring with one oxygen. So this kind of structure, ring structure, is called furanose structure. So if the glucose molecule possesses such a structure, ring structure, then we'll consider it as alpha D glucofuranose. Now the next topic is di and oligosaccharides, where disaccharide means if the sugar molecule is formed with the addition of two monomers, then we consider it as disaccharide. And if up to 10 monomers of sugar are present in a structure, then we call it oligosaccharides. So here I have given one table for your reference, and here on left hand side you will find the names: sucrose, lactose, maltose, trehalose, cellulose, etc. On right hand side you can see the glycosidic linkage present in such molecules. So based on this table, at least you can expect to get one question in entrance examination. Here I have also mentioned the monomers present in these disaccharide molecules. So you need to memorize this table for entrance point of view. So here I would like to explain how glycoside linkages are formed with the help of one or two examples. Here I have chosen the example of lactose. So lactose uh, is formed actually due to the interaction between beta D galactopyranosyl with again one beta D glucopyranose. That means interaction between uh, here you can see this is lactose, which is formed between due to the interaction between beta D galactose with one beta D galactose with beta 1,4 linkage. Let's see uh, the structure. So suppose there is a structure of beta D galactose. So this is a beta structure. So OH group should be on the top of the ring. So we'll be concentrating only on this position right now. Then we have one more beta D glucose. If it is a beta structure, again OH group should be on the top. So this is the OH group. So condensation reaction will take place. So this is the first carbon, second, third, this is the fourth carbon, then again this is the first carbon, second, third, fourth, the fifth carbon, and so on. So here, uh, due to the condensation reaction, water molecule will come out and only oxygen remains in this position like this. So as this bond is formed between first carbon of one sugar molecule with the fourth carbon of the next sugar molecule, adjacent sugar molecule. So that is why this linkage is called 1,4 glycosidic linkage. And as these molecules were in beta form, the linkage started from a beta form of sugar. So that is why we call it beta 1,4 glycosidic linkage. We'll take one more example. This is the example of maltose. So here interaction is between alpha D glucose and alpha D glucose again. So here this is the structure of alpha D glucose. So if it is alpha D, if it is in alpha form, then OH group should be on the bottom. Then again, this is the next sugar molecule. Here also you can find this OH group on the bottom of the first carbon. Then again, this is 2, 3, and this is the fourth one. OH should be on the bottom. So now condensation reaction will take place up here. So it will release water molecule and oxygen will remain. This is the first position, this is the first carbon position. So this will be the shape of the bonding. So as you can see, the bonding is between first carbon of one sugar with the fourth carbon of the next sugar. So this is 1,4 linkage. Then again the linkage actually it took place between two alpha form of sugar. So this is alpha 1,4 linkage. Then the next topic is polysaccharide. Polysaccharide means when the linkage is formed among n numbers of monosaccharides. So there may be two types of polysaccharide: structural polysaccharides and storage polysaccharide. Then again, structural polysaccharides are subdivided into two groups. Uh, it may be present in animal also, it may be present in plants also. So in animal, chitin is one of the structural polysaccharides present in some of the insects and arthropods. So they also act as exoskeleton in arthropods. Then again in plants, cellulose is present on the plant cell wall. Then this is storage form of polysaccharide. So again, storage polysaccharide may be found in animals and also in plants. So in animal, glycogen is the storage polysaccharide and it is stored in liver, and that's a very important point for entrance exam. Then again, uh, starch is the storage form of polysaccharide which is stored in plants. Then again, based on the chain structure of polysaccharide, they may be divided into two categories: linear chain and branch chain. If the molecules forms this kind of chain-like arrangement, then we call it linear polysaccharide. For example, starch and cellulose. I'll show you the structure later on. Then we have branched polysaccharides. Suppose this is a chain of polysaccharide. Then again, you can see the branching in these loops within the chain. So these are branched form of polysaccharides. For example, glycogen, amylopectin, etc. So here in this diagram, you can see on left hand side, I'm showing you the general structure of glycogen, where you can see the branchings are present. And here I have shown you how these actually branching took place. So there are two linear chain. One chain is connected through alpha 1,4 glycosidic linkage. Here you can see alpha 1,4 glycosidic linkage. Then again, you can see alpha 1,4 glycosidic linkage in the next chain also. But these two chains are connected with alpha 1,6 linkage. This is the sixth carbon of this chain on this particular sugar molecule, which is branched with the first carbon of this next chain. So these two chains are interconnected with alpha 1,6 glycosidic linkage. So in the molecular structure of glycogen, you will find both the bonding patterns. You will find both alpha 1,4 glycosidic linkage. Then also you will find alpha 1,6 glycosidic linkage. So here I am showing you one linear structure of polysaccharide. I have shown you the structure of amylose, which is a linear chain polysaccharide, and alpha D glucose are the monomers present in this chain. So these are all connected with alpha 1,4 glycosidic linkage. You can see the glycosidic linkage between first carbon of one sugar molecule with the fourth carbon of the adjacent sugar molecule. Here I have given you one very important table which you should always memorize for your upcoming entrance exams. So I have given you the names of some of the polysaccharides which are commonly asked in entrance exams. Here I have mentioned all the monomers present in these polysaccharides, and also I have mentioned the glycosidic linkages uh, which are found in these structures. And in the fourth column, I have given you the brief idea on the basic structure of all these polysaccharides. So here you can see amylose is a helical kind of chain. Then amylopectin has got one helix with branch with lots of branches. Then glycogen also, it has got same similar kind of patterns, but here the branchings are more as compared to amylopectin. Then starch is a linear form of polysaccharide. Then cellulose is also. So the next point is mutarotation. So here I'm just trying to show you diagrammatically what is mutarotation, and the diagram may not be it may not be scientifically 100% correct, but I just want to show you what is mutarotation with this diagram. So suppose in a test tube, if we take 100% pure saturated solution of alpha D glucose, and if we allow plain polarized light to pass through this test tube, So initially, the light will be deviated to plus 112 degree angle. Then later on, it will reach a stable value of 52.5 degree. Then again, if we take beta D glucose, 100% pure saturated solution of beta D glucose, and again if we allow plain polarized light to pass through this tube, then again initially it will be deviated to 18.7 degree, and later on again it will reach this same stable value of 52 plus 52.5 degree. This specific rotation uh, of plane polarized light is called mutarotation. So here, as per the syllabus, there is no much about mutarotation. So you need to just understand what is mutarotation, and you should just memorize these three values: plus 112 degree for alpha D glucose, then plus 18.7 degree for beta D glucose, and plus 52.5 degree, that is the stable value for both alpha D glucose and beta D glucose. I would also like to mention one more point, uh, that is when starch is hydrolyzed, initially it will be converted into dextrins, then to maltose, and finally to glucose. And the presence of starch may be identified by adding iodine solution. So in presence of iodine solution, starch gives you blue color, intense, it will give you in intense blue color. So here I have mentioned some of the important points which you should revise before appearing for entrance exam. So the points are: sucrose is also known as table sugar, cane sugar, fruit sugar, beet sugar, or saccharose. Then lactose is also known as milk sugar. Maltose is known as malt sugar. Glucose is also known as dextrose. Then heparin is a polysaccharide which acts as anticoagulant, and it is found in the mouth of leech. Hyaluronic acid helps in capacitation of sperms, that means in the activation of sperms during acrosomal reaction. Then trehalose is also known as mycose, and it is produced by some of the bacteria, fungi, plants, and also by some of the invertebrate animals. Then inulin is a polysaccharide of fructose. Here you should not get confused between inulin and insulin. So that is why you need to check the spelling properly. So insulin is a hormone which is produced in the pancreas and which metabolizes sugar. Inulin is a polysaccharide only. It's a polysaccharide of fructose. Then dextrins are intermediates in the hydrolysis of starch. Then fructose is the sweetest sugar. This point is also very important for entrance exam. Then we have some of the sugar substitutes, and these are allulose, aspartame, cyclamate, mogroside, saccharin, sucralose, etc. Out of these many names, the name of saccharin is important. Then glycogen is a multi-branched storage polysaccharide which is found in animal, and the glucose monomers are connected with alpha 1,4 and alpha 1,6 glycoside linkages. And glycogen is stored in liver. This is also important. Then presence of starch, as I have already mentioned in my previous slides, that uh, it may be detected by adding iodine solution. It will give you blue color. So I hope this video will help you in your preparation. Please like, subscribe, and share my channel.